First, we start out with a whole muscle. A whole muscle is then made up of fascicles. Each fascicle is made up of individual muscle fibers. Okay. Action potential arrives at synaptic knob, which then cause calcium channels to open and allowing calcium to move into cell. Increased calcium causes synaptic vesicles filled with acetylcholine to fuse with membrane. Acetylcholine is released from synaptic knob by exocytosis in the synaptic cleft. Acetylcholine is then picked up by receptors on the motor end plate. Acetylcholine is then released back into the synaptic cleft. Acetylcholine is then broken down by acetylcholinease, and choline is then reabsorbed by the synaptic knobs. Choline causes sodium channels to open on the motor end plate, resulting in a creation of a muscle action potential.
The action potential comes down the sarcolemma and stimulates the sarcoplasmic reticulum to open its calcium channels. Calcium is represented by the purple and red skittles. Once the ion channels are open, calcium flows out of the sarcoplasmic reticulum. Mitochondria is represented here. ADP are the yellow skittles, and the mitochondria turns them into ATP, the green skittles. This is the sarcomere. Myosin is represented by the red pipe cleaners. Actin is represented by the pink paper. Tropomyosin is represented by the yellow line. Troponin is represented by the green boxes, and altogether those create the troponin complex. Once calcium is released into the cytoplasm, it binds to the troponin on the troponin complex. Once calcium binds to the troponin, it pulls the tropomyosin off the active site of the actin and allows the myosin to bind to the actin. Myosin uses ATP to power the heads of the myosin to pivot, causing contraction. ATP is used by the calcium pumps to pump calcium back into the sarcoplasmic reticulum. Without calcium bound on troponin, the muscle relaxes.